Sweet taste receptors, known as TAS1R2, TAS1R3, were a groundbreaking discovery by Monell Chemical Senses Center scientists in 2001. These receptors, found in taste bud cells, are responsible for conveying sweetness, but their role extends beyond the taste buds. Recent studies suggest these receptors are also part of a metabolic monitoring system, expressed in gut cells, influencing glucose absorption and uptake. Paul Breslin, PhD, a Monell member and professor of nutritional sciences at Rutgers University, led a team to explore how TAS1R2, TAS1R3 impacts glucose metabolism. They found that both stimulating and inhibiting these receptors can regulate glucose tolerance. Using sucralose, a calorie-free sweetener, and lactosol, a compound that inhibits sweetness, the team observed changes in glucose tolerance during a glucose meal. Also, the novelty of our finding is that the receptor we studied in this experiment affects blood glucose and insulin in different ways during a glucose meal, depending on whether it is stimulated or inhibited, said Breslin. The study involved an oral glucose tolerance test, OGTT, to measure plasma insulin levels and track blood sugar before and after participants consumed a glucose-rich liquid meal. Adding sucralose to the OGTT correlated with an early rise in plasma glucose and increased insulin levels, promoting insulin release. In contrast, lactosol's presence correlated with decreased plasma glucose levels and a slower insulin release. When glucose stimulates the taste receptors before being absorbed into the body, signals are sent through the mouth and gut to regulatory organs like the pancreas, Breslin explained, highlighting the receptor's role in predicting glucose appearance in the blood and aiding glucose absorption. The study suggests that the activity of TAS1R2, TAS1R3 receptors could be linked to a person's health status, as receptor activation levels impact immediate glucose and insulin blood levels. With modern diets rich in high sucrose foods and sweeteners potentially overstimulating these receptors, there's a risk of improper blood glucose regulation, leading to metabolic syndrome and increased risk of heart disease, stroke, and diabetes. Future studies may focus on the therapeutic potential of adjusting TAS1R2, TAS1R3 activity in individuals at risk of metabolic syndrome. Breslin and his team at Monell have shown that sweet taste receptors play a significant role in regulating glucose metabolism, depending on the sweetness of food or drink. Small metabolic changes in a positive direction can bring significant benefits to human life and health when summed over decades and millions of people, Breslin concluded, emphasizing the potential of leveraging TAS1R2, TAS1R3 knowledge to improve dietary health.